everyone, this is Latha Akula. In this video we are going to study about endoplasmic reticulum. So in the previous videos we have completed the plasma membrane and cell wall. Cell wall is present in the plant cells. And inside the plasma membrane there are so many membrane bound cell organelles but present in eukaryotic cells, right? So now we will see these cell organelles one by one. In that the first one is endomembrane system. Here what is the endomembrane system? So there are so many cell organelles were present, right? In that some organelles they may function in coordinated manner. They may be coordinately function. That coordinated manner of organelles which were coordinate with each other are called form the endomembrane system. Those organelles are endoplasmic reticulum, Golgi, lysosomes and vacuoles. These four will coordinate with each other and form the endomembrane system. The remaining mitochondria, chloroplast and peroxisomes, they will not coordinate with endomembrane systems. Hence, they can be separated. They can be studied separately. So, in that endomembrane system, the first one is endoplasmic reticulum. So, this endoplasmic reticulum, which is a network of tiny tubular structures, right? These are the network of tiny tubular structures which are scattered in the cytoplasm. From the nucleus to the plasma membrane, they have been scattered. And this endoplasmic reticulum was first time observed by a person called Garnier. He observed this endoplasmic reticulum first time, but whereas the ultrastructure of the endoplasmic reticulum was first studied under the electron microscope by three scientists. They are Porter, he was very important. The Porter, he studied the ultrastructure. Along with him, Claude and Philip, they studied the ultrastructure of the endoplasmic reticulum. According to them, the endoplasmic reticulum, the ultrastructure of the endoplasmic reticulum, they consist of three parts, three kinds of structures are seen. There was cisternae, vesicles and finally tubules. The first one is the cisternae. Cisternae are long flattened tubular sacs which are interconnected structures present in the cytoplasm, and these cisternae they have been present in the cells which have been actively participated in the protein synthesis. So in which cells they are mainly present? These cisternae are mainly present in the cells which are actively involved in the protein synthesis. In those cells, these cisternae were present. And these are not only there and also present in the some cells which have the synthetic roles such as pancreas and brain cells. In that cells also these cisternae were present. And here in this cisternae, they have a 60th subunit of the ribosomes. They have been attached to this. And next one is the tubules. These are the tubules. They may be the branched or unbranched structures. Actually, these tubules, they have no ribosomes. See, there is no ribosomes present. And these cisternae, they are present in the cells which are involved in the lipid and sterol synthesis. Here, tubules are involved in the cells which have been involved in the lipid and sterol synthesis, whereas cisternae were present in the cells which have been actively participating in the protein synthesis, whereas the tubules are present in tubules. Main function is the lipid and sterol synthesis. The cells which have the major role of lipid and sterol synthesis, in those cells these tubules were present. And the next one is vesicles. So like tubules, these vesicles also do not have any ribosomes. These are oval shaped structures, and these vesicles are abundantly present in the pancreatic cells. In which cells they are mainly present? In the pancreatic cells, these vesicles have been mainly formed, and in spermatocytes, these vesicles are the only endoplasmic reticulum present in them. So, the spermatocytes consist of vesicles only. Next, these endoplasmic reticulum, the compartments that divides the intercellular space into two compartments. One is lumina. So here lumen is nothing but inside the endoplasmic reticulum there is a space is present, right? That is called as lumen. Here this endoplasmic reticulum divides the intercellular space into two compartments. One is luminar compartment and extraluminar compartment. This is the luminar compartment and this is the extraluminar compartment. So the luminar compartment means internal space present inside the endoplasmic reticulum is called as luminar compartment. And what is the extraluminar compartment? Extra means outside. Here the outside the endoplasmic reticulum cytoplasm is present, right? The space within the cytoplasm is called as extraluminar compartment. And depending on the presence or absence of ribosomes, endoplasmic reticulums are of two types. They were smooth endoplasmic reticulum and rough endoplasmic reticulum. The smooth endoplasmic reticulum, see this, the smooth endoplasmic reticulum, they do not have the infrastructure of the smooth endoplasmic reticulum. If you have observed under the electron microscope, the smooth endoplasmic reticulum that appears very smooth surface, with a smooth surface and free of ribosomes. They do not have any ribosomes. See here, this rough endoplasmic reticulum, it is a granular and rough. Whereas smooth endoplasmic reticulum without any ribosomes, it appears smooth. And here, the muscle cells which are rich in smooth endoplasmic reticulum are also called as sarcoplasmic reticulum. So what is the sarcoplasmic reticulum? The muscle cells which consist of smooth endoplasmic reticulum are called as sarcoplasmic reticulum. The functions of this smooth endoplasmic reticulum were, these smooth endoplasmic reticulum, they participate in the lipid and steroid synthesis. Here we have studied here cisternae. Cisternae are present in the smooth endoplasmic reticulum only, and here what functions? Tubules. Tubules are involved in the smooth endoplasmic reticulum only, and here what we have studied, lipid and steroid synthesis, right? That function only we are discussing here. So lipid and steroid synthesis is the main function of the smooth endoplasmic reticulum, and also detoxification of the drugs. Whenever we take the drugs inside, sometimes they may cause some side effects also. Some of the chemical substances present in the drugs may cause side effects, and those drugs detoxification was done by this smooth endoplasmic reticulum, and they help in the muscle contraction and relaxation by uptaking the calcium ions. And here the products of whatever the products, here the rough endoplasmic reticulum that manufactures proteins, right? Those products of rough endoplasmic reticulum, they need to be sent to the Golgi complex. So the Golgi complex that will help in the packaging of ribosomes. Here products of the rough endoplasmic reticulum, they reaches the Golgi complex by means of this smooth endoplasmic reticulum only. Here see, these are the vesicles are present, right? These vesicles are pinched off from the endoplasmic reticulum. They will carry the proteins to the Golgi apparatus. They are nothing but the smooth endoplasmic reticulum only. The vesicles are also. Next one is the rough endoplasmic reticulum. The rough endoplasmic reticulum, when we observe in the electron microscope, how it looks like, it is rough and granular because of the presence of ribosomes, abundant amount of ribosomes. When we observe under the electron microscopes, that give a rough and granular appearance, hence we are calling it as rough endoplasmic reticulum. And here the function of this ribosomes is nothing but the protein synthesis only. And here there are two types of glycoproteins are present in the rough endoplasmic reticulum. They were ribofurine 1 and ribofurine 2. These two glycoproteins, they help in the attachment of 60th subunit of the ABS ribosomes to the endoplasmic reticulum. So here in the endoplasmic reticulum, the ribosomes are attached, right? How they were attached? By means of this ribofurine 1 and ribofurine 2 only. The 60th subunit, that is the largest subunit of the ribosome, will attach it to the endoplasmic reticulum. And what type of the ribosomes were present in the eukaryotes? ATS type of ribosomes only, right? ATS type of ribosomes, the largest subunit is the 60S and the smaller subunit is the 40S. And if you see the functions of this rough endoplasmic reticulum, these are present in the cells which have actively involved in the protein synthesis, and this smooth endoplasmic reticulum is present in the cells which have actively involved in the sterols and lipid synthesis, right? Here these cells area are present in the cells involved in the protein synthesis and secretion, and also this rough endoplasmic reticulum, they provide precursor enzymes for the Golgi complex which involved in the formation of the lysosomes. For the formation of these lysosomes, they are formed from the Golgi complex only. For that, it gives the enzyme precursors to the Golgi complex, and it gives rise to smooth endoplasmic reticulum. That means from the rough endoplasmic reticulum only smooth endoplasmic reticulum develops. So these are the some of the properties of endoplasmic reticulum which are present in the endomembrane system. And the next one is Golgi apparatus. In the next video, we are going to study about the second one in the endomembrane system, that is the Golgi apparatus. And if you like my video, please like, share and subscribe to my channel Latha Akula Biology Tutorial. Please keep watching.